Bismillahirrahmanirrahim. Dear students, Assalamu alaikum and welcome to office.com.pk, which is obviously your very own platform where you come and learn, and we try our best to provide you with the best of knowledge we can. And I hope that we here at office.com.pk are quite successful in that. Now, what is your opinion? How will we know it? For that, I request all of you to please share your views in comment section below, and also I request you to please subscribe to our channel. This will help us know what you are thinking about us, and your any kind of feedback on our videos is going to help boost our morale and our enthusiasm, and we will try to make even better the work that we have started for you. So this was a request that please do subscribe to our channel, because when you see something good on YouTube or anywhere else, it is a moral duty to share your views, your positive or negative feedback about it, so that the person who is doing that thing is able to make it even better. So dear students, in the previous lectures we have been discussing about various theories that were formulated, or various kind of you can say the ideas that were collected from different scientists about how the DNA replication occurs. For your information, I am going to revise what is DNA replication. It is actually the copying of genetic information from one cell to another, because when cell divides, the whole genetic information needs to be copied into the daughter cells. So if DNA replication fails to proceed, or it does not occur properly, then the daughter cells they will be devoid of the genetic information, and the abnormal cells are formed, which are ultimately going to form an abnormal organism. So DNA replication is very much important when it comes to both the mitosis and meiosis, because copying and proper transfer of genetic information is very important, as DNA is involved in the formation of proteins that are going to regulate most of the functions of your body. So we have discussed about the Michelson and Stahl's experiment, and they confirmed from their different experiments that DNA replicates in a semi-conservative manner, and that was very well explained in the previous video. You can check it in the previous video. So what we are going to do today we are going to discuss about the replication process, the mechanism of replication, how the process actually happens, what are the factors that are controlling it and that are making it possible for the DNA to replicate appropriately, so that the genetic information is conserved. Now, what does the word semi-conservative mean? It means that half of the DNA molecule, or one strand of the DNA molecule, is from the parent strand, and the other one is the new strand that is formed complementary according to the parent strand. Now, today's lecture is going to be very important, because DNA replication, this topic is very much important from examination point of view. So you have to keep it in your mind, and you have to listen to me very carefully, and especially the diagram is going to be very important. So you have to be very attentive and very much curious about how to make the diagram, because the diagram is quite tricky, but it is not difficult. Now, the DNA replication, it begins at one or more sites of the DNA molecule. It means that when we have to, or when there is a demand to replicate a particular section of DNA molecule, then in the whole DNA molecule, wherever that section is present or wherever that specific sequence of nucleotide is present, then on all the sites the DNA replication begins. These sites, they have a particular nucleotide sequence. So it means that whenever we have to copy a particular nucleotide sequence, wherever on the whole strand of DNA, wherever that sequence will be present, the replication will start at that all those points. And it does not mean that replication will be at only one point. On all the points replication will start. Now the enzymes that are involved, the most important of the enzyme is the DNA polymerase. The DNA polymerase 3 enzyme, along with other enzymes, they being very important, they start the replication process and they catalyze the addition of nucleotides to the growing strand. So they are going to catalyze the process, as well as they will add nucleotides to the new strand or the growing strand that is going to be formed. And that strand, you all know, it is known as the complementary strand. Now before moving towards the actual replication process or the mechanism of replication, let us have an introduction to the polymerases. What are polymerases? How are they specified for particular process? What are their types? And which one of the polymerases are involved in DNA replication?